Hello, good morning. So, in this session, we will talk about that natural filtration of our body, which filters our blood. That is the glomerulus. Glomerulus. What is the structure of glomerulus? How it works and where it is located? Let us know about all these three about the glomerulus. First of all, glomerulus is a network of anastomosing blood capillaries. It is present inside the kidney of a functional unit, that is a nephron. Nephron inside the kidney. Each kidney has nearly 1 to 1.2 millions of functional units are there. Those are the very microscopic. These are the main functional units. Those are the nephron. So, what is the structure of nephron? The nephron has that is the structure of nephron. So, nephron. This is the Bowman's capsule. Bowman's capsule or Malphizian capsules. This is the PCT. This is the PCT. This one is the DCT. Proximal convoluted tube. Distal convoluted tube. This is a loop of Henle. Loop of Henle. This is the collecting duct. Duct. This is the main functional unit inside the kidney. Inside the kidney, the number of nephrons are, each kidney has nearly 1 to 1.2 millions of these microscopic units. Both kidneys nearly 2.4 millions. So each nephron, at the starting of nephron, a cup-like structure is present. This is called Bowman's capsule. Bowman's capsule. Each Bowman's capsule made with two layers of membrane. Two layers of epithelial membrane. Epithelial membrane. This Bowman's capsule only accommodate the glomerulus. So, at first, before this, the kidneys receives oxygen by a renal artery. This renal artery with oxygen and also has waste materials, loaded with waste materials. When that enters inside the kidney, that is divided into so many arterioles. Okay, each arteriole again divided and comes to the blood capillaries by a afferent arteriole. This is the afferent. This one is the afferent arteriole. This afferent arteriole again divides here number of microscopic. When enter into the Bowman's capsule, this afferent arteriole divides into multiple blood capillaries. That is the look like tuft of capillaries. That is called tuft of blood capillaries that is named as the glomerulus. This one is the glomerulus. Glomerulus means anastomosing units of blood capillaries. So it makes a tuft of blood capillaries that is called glomerulus. Again, all these blood capillaries unite and form a efferent arteriole. Efferent arteriole. Each Bowman's capsule received by a afferent arteriole and leaves by a efferent arteriole. This cup is surrounded by this cup surrounds inside there is a bunch of blood capillaries. Bunch of blood capillaries are anastomosing units of blood capillaries that is called glomerulus. That is called glomerulus. These glomerulus between these between between those blood capillaries there is a connective tissue is present that is called mesengium. That is called mesengium. Mesengium, this is a connective tissue made with uh, mesoglial cells. Mesoglial cells, that is the mesengium. Okay, so this is the structure of glomerulus. Glomerulus means a bunch of blood capillaries which divides into so many. Next, it's surrounds by a covered by a double layered membrane cup-like structure that is the Bowman's capsule. Bowman's capsule. It receives by afferent arterioles and leaves by efferent arteriole. Efferent efferent arteriole, afferent arterioles. Okay, mala how it works? What are the uh, what are the units participate in blood filtration? Here three layers are present. Three layers participate in blood filtration. Blood filtration. What that one is the first one is endothelial cells. Endothelial cells of blood capillaries. Endothelial cells of blood capillaries is blood capillary made with endothelial cells. These endothelial cells as prolifer- uh, proliferated proliferated and has thousands of minute pores. Each cell has thousands of minute pores. The size of those pores are 70 to 100 nanometers. 70 to 900 nanometers. Every blood capillary has endothelial cells, endothelial cell lining. These endothelial cells are proliferated and has so thousands of uh, pores, thousands of holes are present. These holes, size of these holes are 70 to 100 nanometers. Okay, next one is basement membrane. Basement membrane basement membrane here this is the so this is the endothelial endothelial lining. Here middle of this here is a basement membrane. A thick layer is present, that is the basement membrane. This basement membrane made with triple helix polypeptide chain. Triple helix polypeptide type 4 collagen fibers. Type 4 collagen fibers. So when we observe under the microscope, when we observe under the microscope, this basement membrane, middle of basement membrane little bit thick. So this is, it should appear as dense. It should appear as a thickened. That is called lamina densa. Lamina lamina densa. Both edges as, this is lamina externa. This one is lamina interna. Lamina externa, lamina interna. Middle of this lamina densa. This lamina densa, this base membrane, uh, at the edges side, that is translucent, but middle it will be very thick. It is made with collagen type 4 fibers. Next, this basement membrane electronegatively charged. That one is the second one. Next, third one is visceral epithelial cells. Visceral epithelial cells, these Bowman's capsules made with two layers. One is the visceral epithelial cells. Next, this one is the parietal epithelial cells. These, the cells of visceral epithelial cells has food processes. Food processes. These are called podocytes. These are called podocytes. Podocytes. With the help of these food processes, they attach to the basement membrane. And also these podocytes has so many finger-like processes, finger-like projections are present. All these finger-like projections are interdigitated. Like this, their fingers are interdigitated between. Because of that interdigitation, there will be slit appears, that is called filtration slit. Slits. Filtration filtration slits because of interdigitation of those finger-like projections. The between these those between those finger-like processes, there is a slits appears. Those slit is called filtration slit. That is the size nearly 20 to 30 nanometers. 20 to 30 nanometers. 20 to 30 here is 7. They filters 70 to 100 nanometers. Again, when you come to here, that slits is only about the 20 to 30 nanometers. Okay, so this uh, this is the structure of the filtration unit. Next, how the filtration process will takes place means, so these afferent arteriole, this is the lumen, this is the wider and more lumen. So when you compare to the efferent arteriole has low, um the lumen space is narrow, and lumen space is very low, and it is a narrow outlet. Because of that, the pressure exerts inside the blood capillaries. Again, hydrostatic pressure of those glomerulus. Next one is each here unusual setting is each every organ reaches by a uh, arterioles and lives by venules, but here both is arterioles because it helps to uh, put a pressure, a great pressure inside the glomerulus. Again, these blood capillaries supported by the mesangial cells, that is the mesoglia. These mesoglial cells has a one characteristics. They has a contractile cells, the contractile cells. Because of that contractile characteristics, they also helps in blood filtration. This is the filtration. So totally this is the filtration process is depends upon the size barrier, and depending upon the size only the filters. And also charged ions, both these endothelial cells, basement membrane, visceral epithelial cells, these podocytes, all these three are negatively charged means they attract the positive ion charge, positive ion charged cells. Because of that, they uh, the positive ion cells are easily filtered. So when you see the blood has along with blood cells and protein substances, next to plasma proteins and blood cells, along with the waste materials, along with these digested substances, that is the glucose, amino acids. All these glucose amino acids are filtered very easily because they are very small size. And also creatinine, urea also filtered, but they don't allow to filter the blood cells and plasma proteins. Why? Because those size are very bigger when they compare to the uh, these size of these pores. These pores, this is the process of blood filtration. This is a natural filtration in our body inside the kidneys, in the premises of the nephron. Okay, so here you have to remember endothelial cells, next basement membrane, visceral epithelial cells, and mesangium. mesangium The mesangium made with the connective cells, that is the mesoglial cells. These are the contractile cells and phagocyte cells, and also proliferate cells. Proliferation అవసరానికి తగ్గట్టు అవి మార్పు చెందుతూ ఉంటాయి. మరి ఫాగోసైట్స్ ఎనీ అన్వాంటెడ్ సబ్స్టెన్సెస్ హియర్ దే engulf అంటే తినేస్తాయి. నెక్స్ట్ వన్ ఇస్ ద contractile cells. At the time of blood filtration, they contract and helps to the blood filtration process. Okay, next one is the after filtration, the along with water which they are present here, that is called primary urine. Next, these filtration primary urine along with this PCT, DCT, loop of Henle. Next one is the collecting duct. They concentrate and which are useful. Again, they uh, absorbs and secretion by the all totally the urine formation will takes place. This is the structure of glomerulus and functions. Okay, thank you.